Hello and welcome back to this new lesson. I'm talking to you about interactions between viruses and cells and the different kinds of infections we have. So, first of all, the virus needs to recognize the cell, the potential host cell, and to bind to it. The binding is permitted thanks to the presence of glycoproteins on the surface both of the cell and of the virus. Glycoproteins are called receptors when talking about cellular glycoproteins and antireceptors when talking about viral glycoproteins. The interaction between antireceptor and receptor is really specific. Consider that on the surface of the cell we have primary receptors which have a very high affinity for the antireceptors, but we also have co-receptors, also called secondary receptors, which have a lower affinity for viral antireceptors, but they are essential to permit a better adsorption. What I mean by adsorption? The adsorption is the specific recognition that occurs in between viral antireceptor and cellular receptors. So adsorption is the first step of any infection. The second step is penetration. Penetration may occur in different ways. Basically, we have external fusion and internal fusion. External fusion is characteristic of coated viruses. That is, viruses which have pericapsid. So, what we have is the fusion of the pericapsid on the surface of the cell and the release of the capsid and the viral genome inside the cytosol or directly inside the nucleus. The external fusion occurs thanks to the presence of fusions proteins which work better in a neutral pH. On the contrary, the internal fusion occurs better in an acid pH. What I mean by internal fusion? Internal fusion is the enter of the virus thanks to endocytosis vesicles. So the fusion occurs between the virus and the vesicles membrane. After penetration, we have the exposure of the virus's genome, either in the cytosol or inside the nucleus. Then we have the genome replication. I talked to you about it in the last lesson. After the replication, we have the assemblance of the new viral progeny inside the nucleus or inside the cytosol. Then we have a very important step, which is the maturation. The maturation is essential for the new progeny to be able to infect other cells. Maturation may occur inside the nucleus, inside the cells, inside the cytosol, or at the cellular membrane, or directly externally. The last step of an infection is the exocytosis, or the release of the new progeny externally. The release of the new progeny may occur by killing the cell or without killing the cell. The best way for the virus to survive is to let the cell be functional. So when the cell dies, the virus is not able to replicate anymore. As I told you, we have different kinds of infections. The first infection I want to talk to you about is the abortive infection. it means that the virus is not able to enter the cell, and so it's not able to replicate. This may occur whether the virus is defective. What I mean by defective? By defective, I mean that the virus is not well matured, so it's structurally completed, but it's not able to infect new cells. Or if the cell is not sensible. A sensible cell is a cell which has the right receptors on the surface. A cell may be sensible but not permissive. A cell is permissive when it's able to provide the virus with all the metabolic pathways, all the energies, all the molecules, and all the structures it needs. So a cell can be sensible and not permissive. It can be not sensible but permissive. It can be non-sensible and non-permissive, but it can be also both sensible and permissive. When a cell is both sensible and permissive, then we have a normal viral infection. An infection may be productive when the cell is both sensible and permissive. An infection may be non-productive when the cell is sensible but it's not permissive. We talk about cytolytic infection when the release of the new viral progeny gets the cell to die by a cytolytic mechanism. An infection is non-cytolytic if the cell can survive. Finally, an infection is persistent if the cell survives and the virus is able to replicate itself constantly. Actually, we have different kinds of persistent infection. The latent infection, when the cell is permissive only in specific times of the cell cycle. Chronic infection, when the virus is always able to replicate itself throughout the cell cycle. An infection is slow when it has a very long latent time followed by a proper viral replication. So that's all for now. I'll see you in the next lesson. Bye!